Okay, so now we'll talk about deletion mapping. So now when Benzer, he had large number of mutations, and if he went on doing those kind of 2-point crosses, it would take a very long time. It's not that it's impossible, but it would take a very long time. But ingenious that genius that Benzer was, what he did, he isolated several deletion mutants. Now deletion mutants are those mutations in which a part of the gene is deleted. Now, if a part of the gene is deleted, we know that that cannot be reversed back, okay? And that will not complement with any other mutation that is making that particular deleted region homozygous, okay? So that is the crux of deletion mapping. It looks difficult or it looks very hard, but otherwise once you understand the principle of it, it's very easy. Now just to make clear first, and then we'll talk about how Benzer did. Uh, like say, suppose if this region is deleted over here, this is on one homologue is deleted, and if the second homologue has a point mutation at this region which is overlapping with the deleted region, so therefore here you will not see any complementation because there is a loss of homozygous region. There is a homozygosity is lost. However, if you have a deletion over here, but if there's a point mutation outside the deleted area, so then you will see complementation. Okay, so yes, you will see complementation. Suppose if there are two deletion mutants, and if those deletion mutants, these are overlapping over here, then you will not see any complementation because both the regions are actually lost. However, if you have one deletion here and the other deletion beside it, that means you have certain regions which are there are regions which are not common in either of the deletions. You see complementation. Now, when you have this condition also, there is also a possibility of recombination, where if you have recombination in this region, you will get a wild type wild type chromosome. So you can do you can get recombinants of the wild type progeny. Now, what Benzer did was he used these deletion mapping. Okay. Now, for doing this analysis, first he isolated several deletion mutants, and then he did this kind of screening. When he did this kind of screening, what he could do was he could characterized 7 deletions which are large deletions, and he called them as the big 7. Okay, he called them as big 7 deletion, and these big 7 deletions, they have a particular name. I have not written the names, but you can find the names in the books. Okay, now these deletions, what they did, they, with the help of these deletions, he could divide the entire R2 locus into 7 different regions from A1, A2, A3, A4, A5, A6 and B. Now what you see over here is that A region is very large and B region is small, and this is exactly what Benzer got. He got A2 was almost double the size of the B locus, right? Now these large deletions or these regions, they were further divided into small subsections using another set of deletions. Okay, so as a result, this interval was subdivided into, there were sub-intervals, and they range from A1A to B10. Now if I show you the image in the book, you can see this image in any book, but this is Simmons, and you can see over here that these are the sub-deletions and those are the large deletions. Now if, let's say, how do you map using deletions? Now if you suppose if there's a mutation which does not complement with A1, A2, A3, A4, A5, okay, till A5 it does not complement, but it complements with A6 and B. So what it means is the mutation is falling somewhere in this region, because it's falling in this region from the distance from A5 to A6, because if the mutation was here, it would have complemented with A5. If the mutation was here, it would have complemented with A4 or these. But because it is not complementing with any of these deletions, it's only complementing with this deletion, which means that the mutation is in this range. So therefore, if if it's complementing with A6 or this deletion, so it's very natural that it will complement with this. Okay, so this is how. So next, what Benzer did was he took now mutants of this region, A6 region, and finally you could very precisely point, or he could map that particular mutation where it was present. So using this kind of strategy, deletion as well as combination or complementation, recombination, he mapped the entire genome. Okay, so therefore this is called as fine structure analysis, genetic map. And this is a genetic map because I think you all should know that these mapping, the map which is made by doing genetic crosses, it's called genetic map, and the map that's created by the DNA sequencing, that's a physical map. Okay, now to end it, I think I'll give you do a numerical as always. Now here there is a region A and B, okay, and these are the different regions which have been deleted. So there are 5 deletions, R, S, T, U, V, okay. I've shown the extent of deletion, and based on these deletions, the entire region has been subdivided into A1, A2, A3, A4, B1, B2, B3, B4 and B5, okay. Now the question that follows this is, first question is, in which gene does a mutation occur if it complements with U but does not complement with S? In which gene does a mutation occur if it complements with U but it does not complement with S? The second is, where is the mutation located if it does not complement with S or V? The third is, where is the mutation located if it complements with T? Fourth is, if a mutation is in gene R, will it complement with S or V, both or neither? Fifth is, can a mutation complement with V but not with U? Sixth is, give the most probable position of a point mutation if it recombines with deletions S and V but not with the other 3 deletions. And the last is, give the most probable position of a point mutation if it does not complement with V, does not recombine with R, but does recombine with the other 4 deletions. So these are the questions based on this map. So I would like if you all do it. There's a second question which I'll just write over here. This is an easy one. This is in Drosophila, the 2 recessive mutants B and E. This is black and this is ebony. Black and ebony, they are both recessive mutations and they give rise to black body, which is unlike the wild type, it's grey. Okay, and these both are black. Now gene mapping analysis has shown this to be on chromosome number 2 and this to be on chromosome number 3. Now what scientists have done is they have crossed B and E and they have found the heterozygous to be all grey. Okay. Now the question is, knowing the information about the gene mapping, was it necessary to perform the complementation test? Okay, because the complementation test, when it was done, you got grey. So the question is, knowing this information that they are on 2 different chromosomes, was doing complementation necessary? So your answer is, if you say yes, you have to say why. If you say no, you have to say why. So this is all for the gene mapping part. And so with this, I have completed the evolution of that topic of evolution of gene, the concept of evolution of gene. And then the next class, we'll talk the next, we'll see what, I think the next topic I'll decide and I'll let you know. Thank you so much. Do the numericals and I'll give you more numericals. Thank you.